daughter cells are produced,
Yes, what?
और उनका genetic material, two daughter cells are produced in mitosis,
Mm-hmm.
and in meiosis, four daughter cells are produced.
Okay. Any other specific difference?
And the genetic information in mitosis, the parent cell and the daughter cell has the same number of chromosomes, and in
Mhm.
meiosis, the number of chromosomes reduces to half.
Okay. Which I want some other one to respond. Um, or you can also write in chat. Which of them is responsible for division of somatic cell, and which one is responsible for division of reproductive cell?
रिप्रोडक्टिव के लिए मियोसिस है और माइटोसिस दूसरे वाले का।
Very good. So these are the major differences between mitosis and meiosis. And in mitosis, it is not only the number of chromosome remains same, the genetic material also remains same. There is no variation, there is no crossing over, there is no new combination of genes produced in mitosis. While in meiosis, new combination of genes are produced. So in exam, even if it is like, for example, there are 5 keywords given to you, 5 pairs are given to you, and we ask you differences, you are simply not writing one difference. You are writing 2 till 3 logical differences. Whatever the differences discussed right now, I think most important were that mitosis is the division of somatic cell, meiosis is the division of reproductive cell. In mitosis, the number of chromosomes remain the same as that of parent cell. In meiosis, at the end, the number of chromosomes reduced to half. In mitosis, there is the genetic material remain exactly the same. There is no crossing over. There is no new combination of genes produced. And in meiosis, crossing over take place. New combination of genes are produced. If someone write at the end of mitosis, two daughter cell produced, at the end of meiosis, four daughter cell produced, this is also correct. But the first 3 were the major differences between mitosis and meiosis. Okay. In your miscellaneous section, I not only uploaded practice material as well as the relevant marking key, so you can refer it as per your ease. Okay. Now, question number 2, why do homologous chromosome pair in meiosis synapse but not in mitosis? Why like crossing over is happening? What will be the result of this? Why homologous pair chromosomes are pairing up in meiosis 1? Yes. Come on. This is mitosis 1. Okay, it's wait. Here is the possibility. They are saying why homologous pair chromosome in meiosis 1. In prophase, we cannot see there is no pairing up of chromosome, right? The whole chromosome, the two separate chromosomes are aligning and are aligning themselves at the equator. While in meiosis 1, homologous pair of chromosomes are pairing up. Homologous pair of chromosomes are formed after the end of meiosis 1. Do you think of meiosis the number of chromosomes are reducing to half?
Yes.
Hello, are you listening? Please respond so I can understand whether you can get it or not. While
Yes,
in me, yes, beta.
but in meiosis, meiosis is used for reproduction, right? So that's why the
Yes,
homologous chromosomes pair up, and then you told कि वो divide मतलब end में वो separate भी होते हैं.
yes. In
Yes,
meiosis, homologous pair of chromosome arrange so that they are reduced to half. The homologous chromosome do not pair up. Homologous chromosomes are separating from each other. The whole set of chromosome is going on the other side. While in later stages of mitosis, the centromere is breaking. Did you get it? Did you get it or not, beta?
मिस आप वही कह रही है ना कि जब meiosis होता है तो हम रिडक्शनल फेज के थ्रू जाते हैं, बट इन माइटोसिस जो नंबर ऑफ क्रोमोसोम्स होते हैं वो सेम ही रहते हैं, बट इन रिडक्शनल फेज उनको पैर अप होना होता है ताकि फिर वो नंबर हाफ हो जाए इन द एंड.
Yes. Okay, now let's see what is the logical way to say pairing allows when once the homologous chromosome pair up, first of all, it creates a chance of crossing over so that new combination of genes are produced, and it also ensures that each daughter cell receive exactly one pair of chromosome having the total number. Okay, while like if the question was both about mitosis and meiosis, so you can also say in meiosis no crossing over required, no new combination have to be formed, and no reduction division is required. That's why there is no need of there is no need of pairing of homologous chromosome. So you need to say both the things. I hope I made it clear. Okay, then
Yes. मिस ये जो
question number,
डॉट सेल वाला था वो आप बता सकती हैं दोबारा?
which one?
जो भी आप उसका आंसर दिखाई थी उसमें से डॉटर सेल वाला था.
Yes, I can show you. I can share it. I said when homologous pair of chromosomes are formed, it creates an opportunity for the chromosome to exchange their segments, crossing over take place, new combination of gene is produced. Pairing of homologous chromosome allows genetic variation. If you can see the diagram, sorry, beta. You can see the diagram, you can see after crossing over, the chromosomes have exchanged their segments, means new combination of gene is produced. So pairing of homologous chromosome is giving us two advantages. Number one, new combination of gene is produced in meiosis, and the in the later stages of meiosis, it facilitates the process of reduction division. From the homologous pair, one set of chromosome move on the opposite pole, the other set of homologous chromosome move on the opposite pole. As a result, the number of chromosome reduced to half. Am I clear?
Yeah.
Okay. Then question number 3. I remember while we were doing this topic, we were trying to conclude. So some of you were still confused till the end. So purposefully, I created this worksheet so everyone understand from this concept what are the logical things we should keep very clear. Okay, if a cell we know that before the cell undergo the division, it undergoes the interphase, the resting phase. Now, examiner can pick any of the phase. Like if G1 phase get skipped, if S phase get skipped, if G2 phase get skipped, so what will be the consequences? Here the question is, if a cell skipped S phase before meiosis, what would happen? Whether mitosis or meiosis, if synthesis did not happen. So now what will happen? What is the purpose of S phase? What is the purpose of S phase? What does S phase serve?
To prepare for the division,
Yes, what?
to prepare for the cell division, to prepare the cell for the cell division.
Identical
Synthesis refer to replication of DNA. See, this is from the cell phase. Keywords matter a lot in biology. In S phase, what exactly is happening? In S phase, the signals are received for the cell to divide again. DNA replicates. It means that, for example, if it was a human DNA, if it was a human cell, so there will be 46 chromosomes. Their pairs will not be formed. This is meiosis. I can see 1, 2, 3, 4, 5, 6. 6 chromosomes, and they have their sister chromatid. Can you see? The blue chromosome appear double, the red chromosome appear double. Every chromosome is appearing double. So when, at which stage this doubling had taken place? When did this doubling had taken place?
In the S phase,
In S phase. What if S phase did not happen? So will be sister chromatid will not be available for further division in meiosis 2. See, in meiosis 2, the sister chromatids are separating from each other, right? So S phase is really, really very important because it helps the cell to make the sister chromatids. Let me show you the exact marking key as well. Okay, I will see. Okay, the cell would not have sister chromatid. It would likely to fail to divide. This, if someone write the cell would further not be able to divide, this is the final consequence. But what is the actual reason? The cell is not going to have the sister chromatid. Okay, so the process of meiosis will not be continued. Am I clear? I'm trying my level best to discuss each and every question in detail so you understand it properly. Okay, why must G2 happen after S phase but before mitosis? Any question which is specific to mitosis, or it means that the similar question can be asked related to meiosis as well. This question is related to cell cycle. This is the diagram of cell cycle. You must understand it, learn the sequence. They are saying why why G2 phase must happen after S phase but before M phase. For that, we need to seek what happens in G2. In G2, the cell continue to grow, new DNA checked, and any error repaired. Preparations are made for cell division to happen. Yes, Saira, you want to answer?
मीन्स बिकॉज इन एस फेज आपका जो डीएनए होता है इन माइटोसिस स्पेसिफिकली वो रेप्लिकेट होता है, तो जब वो रेप्लिकेट होगा उसी के बाद जी टू फेज में जाके हमें पता चलेगा ना कि मतलब उसमें जी टू उसमें देखेगा कोई एरर है या कुछ है, तो उसको रिपेयर करना होगा, फिर उसके बाद ही माइटोसिस में फर्दर वो जा पाएगा.
Error have to be removed. So error must not move on to the cell where error must not pass on to the cell that is dividing. Is that clear?
Yes, miss.
Okay. Now, you know that similar topic can be asked for the from the perspective of short question. We might give two terminology, and we might ask you to differentiate between them. We might um, give you the structures to identify that which state does it belong to. I'll try to create one more worksheet in which I'll show you different pictures of photomicrograph where you will try to identify the stages of the cell cycle. Okay, the stages of cell division. Okay, there are some very important MCQs. Many of the students were like confused that as sometime the language of the question is tricky. So purposefully, I made high thinking order questions for mitosis and meiosis, and I'm presenting it to you. So let's see how you respond to it. A drug prevent the formation of a spindle apparatus. You know what is a spindle fiber, right? A person has consumed certain type of chemical substance, a drug, which is stopping the spindle fiber formation. Which stage of mitosis is directly blocked? Okay, the question is related to mitosis. It means that you must have a grip on the diagram, on the concept, right? This is mitosis. Okay, the person has consumed a drug which stop the formation of a spindle fiber. These are spindle fiber, right? These blue are spindle fiber. If spindle formation is stopped, now they are asking which I'm sorry, which stage of mitosis is directly blocked. Oh, I got answers in chat. When do the spindle formation starts? When do the spindle formation starts? In prophase. Spindles are formed from microtubule. Okay, so now we have a confusion that whether it is prophase or whether it is metaphase. In the description of prophase, it was arrangement of
मीटिंग तो ज्वाइन कर लो आपने मीटिंग ज्वाइन नहीं करी
so in prophase the centriole are moved to the opposite pole, and spindle formation actually occurs in metaphase. So metaphase will be stopped. Next is independent assortment occur during metaphase 1. Okay, before I go into the detail of this question, I want to show you this diagram. Okay, these are homologous pair of chromosome. The homologous pair of chromosome are arranged at equator. And now, if you look carefully, from the homologous pair of chromosome, one set of chromosome move on the opposite side and the other set of chromosome move on the opposite side. Is it for sure that this particular chromosome will move on this side and this particular chromosome will move in the opposite side? Can the attachment of a spindle fiber vary it? Is it necessary that, for example, let's suppose blue is the paternal chromosome. Is it for sure that paternal chromosome, all the blue paternal chromosome will go in one cell and all the red maternal chromosome will go in the other cell? Is there any such restriction or it is at random?
No restriction.
There is no restriction. This is called independent assortment. What is independent assortment? In meiosis, first of all, homologous pair of chromosome are formed, and during anaphase, when the homologous pair of chromosomes are separating, it is not sure, it is not restricted. There is no such specific criteria that whether the paternal chromosome will move in one cell, maybe two paternal chromosome and one maternal chromosome, maybe two maternal chromosome and one paternal chromosome move in the opposite pole. This uh, this randomness is called independent assortment. Like there is no fixed rule that which of the chromosome have to move. Now you got independent assortment idea. Independent assortment occur during metaphase 1. This is primary reason why sibling look different despite having same parents. Cancer cell divide rapidly. Skin cell replace themselves every few weeks. DNA replicate during S phase. What makes the most logical explanation for this statement?
A.
A.
The sibling look different, even they have the same parents, right? Even identical twin look different. Okay, this is a calculation question. I want you to concentrate. I hope everybody is getting it. 2 or 3 students are responding, few are writing in chat. I really appreciate it. And I want, don't think your answer is correct or incorrect. If it will be incorrect, it will be corrected. And I want all of you to participate actively. Okay, look at question number 3. Yes.
It's 48
sorry.
or 5?
A gorilla has 48 chromosomes. How many chromatids are present in each cell during metaphase 2?
24.
Miss,
Yes, Peter.
metaphase 2 is related to meiosis stages, right?
Yes.
So the number of chromosomes became half.
In meiosis 2, this is meiosis 2. In meiosis 2, the number of chromatids are like they are definitely half from the starting. In starting, they were double. The chromatids were double, and in meiosis, the chromatids are decreasing. For example, if I count 1, 2, 3, 4, 5, 6, 7, 8, these are 8 chromatids. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There are 12 chromatids. And in meiosis 2, the number of chromatids are 1, 2, 3, 4, 5, 6. The number of chromatids are reduced to so what will be the answer? What is half of 48?
24.
24.
Yes, the number of chromatids will reduce to half. Which process is most responsible for infinite variety of genetic combination in human offspring? The doubling of DNA, the crossing over in prophase 1, the separation of sister chromatids in mitosis, or cytokinesis. Like, which of the following brings variation among the offsprings?
Miss, शायद A.
Okay, if you write your answer A or B, mention question number because it's difficult for me to catch up. I hope you are okay. Yes, it is crossing over. It's not A. A is ensuring there is genetic material is double. Okay, not C, beta. What is happening in C? The separation of sister chromatids in mitosis. In mitosis, genetic variation do not take place. At the end of mitosis, no genetic variation take place. The starting genetic material and the ending genetic material are exactly the same. C is not the answer, Rita. Am I clear? Anyone who wrote answer C is incorrect. And I hope you got it. Savira, you got it, beta? Okay. Non-destruct. Yes. Yes, Saira. You want to
Yes,
ask
क्रॉसिंग
something? Yes,
यस मिस
please.
क्रॉसिंग ओवर इसीलिए होगा ना क्योंकि जब एस फेज होता है तो वो जो डीएनए होता है वो डबल हो जाता है बट जो मतलब वेरिएशन आती है वो तब आती है जब क्रॉस ओवर हो रहा होता है
S phase happen in both in mitosis and meiosis, right?
यस यस
S phase is not bringing variation. When homologous chromosome did crossover and exchange their segment, it is the cause of variation. Is that clear? Shall I continue the next one? Okay. Non-disjunction is an error where chromosomes fail to separate. If this happens during meiosis 2, the resulting gametes will have, okay, we showed you a proper stage of meiosis. Non-disjunction is a kind of error where chromosomes fail to separate. Okay, for example, this whole bunch move on this side, and this set of chromosome, this side of the cell has one less chromosome. Means, if I have my writing tool. Okay, so what if there is one extra pair move in this side and this chromosome is removed? Like on this side, there is one less chromosome, on the other side, there is one extra chromosome. This kind of error is called as non-disjunction. I told you two new terms today. The first one was independent assortment, and the second one I'm telling you is the non-disjunction. Non-disjunction is a condition in which even the question is self-explanatory. The examiner has given you the explanation of the word. Non-disjunction is an error where chromosomes fail to separate. This error happened during meiosis 2, resulting gametes will all normal chromosome number, double amount of DNA, one gamete n plus 1, the other gamete n minus 1, and two normal. Like in meiosis, 4 daughter cell have to be formed, right? Or all 4 gametes will have n plus 1. So what do you think? What will be the possibility?
C.
Because why B? Are you talking about B or what?
C. C.
C. Okay. What if this type of error occur? So in in the later stages, in the later stages, this cell will have one extra chromosome. This cell will have one less chromosome. And when it will divide further, two, one cell will have n plus 1, one cell will have n minus 1, and two cell will have exactly the same number of chromosomes. So this is the correct answer. C is the correct answer. Is it clear to everyone? Is this clear to everyone?
Yes.
How does anaphase of mitosis differ from anaphase of meiosis?
इसका आंसर डी है.
Okay, let's have a look. This is anaphase of meiosis. Okay, the pair of chromosomes are separating, and this is the anaphase of mitosis, where the sister chromatids are separating, the centromere is breaking. Okay, let's see what examiner is asking. Okay, in anaphase, mitosis separates sister chromatids. In meiosis 1, it separates homologous pair of chromosome. In mitosis, there are no homologous chromosome. There is no difference. This is incorrect. Mitosis result in haploid cell. Meiosis result in diploid cell. This is incorrect statement. So what is your suggestion? Yes, very good. Very positive attitude today. Alhamdulillah. Okay. Meeting, we have less than 1 minute. No worries. We will rejoin in 5 minutes from the same link. A researcher observed a cell with a cleavage furrow forming. This cell is most likely